In every living cell, DNA contains the genetic blueprint for the organism. This blueprint must be replicated accurately, repaired when damaged, and recombined to ensure genetic diversity. These three processes, DNA replication, repair, and recombination, are fundamental to life itself. In this video, we will explore each of these processes in detail, highlighting the molecular players involved, the specific steps, and the importance of these mechanisms for cell survival and function. DNA replication is the process by which a cell makes an identical copy of its genome before cell division. It's an extraordinary complex and precisely regulated process. Let's examine the steps involved in DNA replication, focusing on the molecules, inhibitors, and the intricate mechanisms at play. Our first step is the initiation of replication. Replication begins at specific sites known as the origin of replication. In prokaryotes, there is generally a single origin, while in eukaryotes, multiple origins are required to replicate the much larger genomes. The initiator protein in prokaryotes is DNA, while in eukaryotes, the origin recognition complex (ORC) marks the origin site. Once the origin is identified, Helicase binds and unwinds the DNA, breaking the hydrogen bonds between complementary bases. The MCM complex, a helicase in eukaryotes, is activated by phosphorylation by cyclin-dependent kinase (CDKs). Here also inhibitors works. Certain molecules like caffeine and specific kinase inhibitors target CDKs, preventing the activation of the origin recognition complex and in turn inhibiting the initiation of the DNA replication. Our second step is the DNA unwinding and stabilization. As helicase unwinds the DNA, it generates positive supercoiling ahead of the replication fork. To elevate this strain, topoisomerase enzymes comes into play. Topoisomerase 1 creates transist single strand breaks, while topoisomerase 2 introduces double strand breaks to resolve taurosinol in strain. Here, the SSBs, single strand binding proteins, stabilize the unbound single strands, preventing them from re-annealing before replication can proceed. Next is the third step, RNA primer synthesis and DNA elongation. DNA polymerase cannot start a new strand of DNA on its own. It requires a primer. The enzyme primase synthesis short RNA primers on the single-stranded template DNA. These primers provide the 3-hydroxyl group required by DNA polymerase to begin adding nucleotides. In prokaryotes, DNA polymerase III is the primary enzyme responsible for synthesizing the new DNA strand, while in eukaryotes, DNA polymerase delta and DNA polymerase epsilon perform this task. Our fourth step is about the elongation of the leading and the lagging strands. Replication proceeds continuously on the leading strand as the DNA polymerase moves in the same direction as the replication fork. On the lagging strand, however, the DNA is synthesized in short segments called the Okazaki fragment. These fragments are later joined together via the enzyme Helicase. Our fifth step is about the replication termination. Replication terminates when two replication forks meet. In eukaryotic cell, replication at the end of chromosomes called telomeres requires the action of the enzyme telomerase. Replication terminates when two replication forks meet. In eukaryotic cells, replication at the ends of chromosomes called telomeres requires the action of the enzyme telomerase. Telomerase extends the telomeres to prevent the loss of essential genetic information during successive rounds of replication. DNA damage occurs consistently within the cell from both endogenous and exogenous sources. The integrity of the genetic material must be maintained to ensure proper cellular function and organizational development. Let's explore how DNA repairs work at the molecular level.